Hi, everybody, and thank you. Welcome to a double feature from the Avian Ecology Lab. Our first speaker today is going to be Rosa Ponce Vargas. Rosa is from North Carolina, and she attended the University of North Carolina at Wilmington with a biology and a degree in biology. She had lots of different experience as an undergrad working in really diverse taxa, from terrapins, marine mammals, butterflies, shellfish, and birds. It was really in her undergrad that she developed an interest in birds, and she focused some of her postgraduate experience on birds themselves. And her first real postgraduate job was working at Hastings Reservation in California, where she worked on the acorn woodpecker, another cooperative breeder. So she learned a lot of the techniques of studying cooperative breeders. She also worked with the CTT grid, the same kind of tracking grid that we use here. She went on to Hawaii, where she actually worked on mosquitoes, but in fact, in the broader context of how climate change might be affecting mosquito range and the impact of disease on endemic birds. So one of the things we've always talked about in hiring interns is looking for a well-rounded team, a team that gets together, that really gets along together and works together well. And one of the things that really impressed us about Rosa was she just won a slew of awards during her senior year. And it was in all different things, contributions to the community, sustainability, as an advocate for social justice and human rights. So she just seemed like a great team player. And that was absolutely writing on the wall. Rosa was a fantastic team player. She was optimistic all the time and cheerful. She just took this tremendous amount of pleasure being out in the field, and we always look forward to her daily observations from the field, something that she'd see. She'd text us all about it, and they were fun. So she also is a total ping pong hustler. Don't bet any money with her. So with that, I'm going to introduce Rosa. She's going to talk a little bit about helper effects, which you'd think we know everything about it, but Rosa's continuing to educate us. Rosa?
All right, thank you so much, Reed. So with that, my name is Rosa, and the title of my presentation is Compensatory or Additive Helper Effects on Food Provisioning and the Cooperatively Breeding Florida Scrub Jay. So a little bit of background, what is cooperative breeding? Cooperative breeding is a reproductive system in which non-breeding individuals, also known as helpers, assist breeders with raising current offspring. Some benefits of the system include an increased survival rate and increased reproductive rate. And this occurs in 18.5% of birds, which includes our very own Florida scrub jay. If groups have helpers, they can have up to 6, and these helpers remain at home until they find a territory for themselves. Until then, they assist with territory defense and some breeding-related activities, including feeding nestlings and young fledglings. So how do helpers affect breeder investments? When helpers are present, breeders may modify reproductive investments to either minimize the cost of producing offspring or maximize offspring survival. If breeders are able to reduce their investments relative to any additional care given by helpers, and if breeders are the ones benefiting from the strategy, it is known as a compensatory care strategy. If helpers are able to improve overall care and breeders do not reduce their own care, and juveniles are the ones benefiting, it is known as an additive care strategy. So what factors influence investment strategy choice? There's a variety of factors that can influence which care strategy breeders choose to implement, but the ones that I'll be focusing on is brood size, which is the number of young per breeding attempt, and I'll also be focusing on the number of helpers. So the question I wanted to ask is, how do helpers influence the rate of breeder male food provisioning to post-fledgling juveniles? I hypothesize that helpers will increase the overall number of feeds to juveniles, And based on that, I predicted that breeder males will implement an additive care strategy. So for my methods, once again, my study organism is the Florida scrub jay, and each individual was identifiable by their unique color band combinations. My study site took place at Archbold Biological Station, which is composed of theromorphic scrub and flatwoods, which the Florida scrub jay needs to thrive. And for my experimental design, I visited 24 predetermined groups, 10 groups which were composed of only a breeding pair, 8 groups that were composed of a breeding pair and 1 helper, and 6 that were composed of a breeding pair and 2 or more helpers. These groups were observed 2 weeks after their fledge states, and when I visited each group, I conducted a 2-hour-long focal feeding observation where I measured male, helper, and total feeding rates. So, starting off with my results, the first thing I wanted to ask was, do breeder male feeds change with the number of young? And here you can see that although there was a slight positive trend, overall, the breeder male feeds did not change with increasing brood size. And this is reinforced by our non-significant p-value. So that led me to ask the next question of, do breeder male feeds change with the number of helpers? And here you can see that if no helpers were present, the number of breeder male feeds increased a lot. But once you started looking at groups that had 1 helper or 2 and more helpers, you can see that the number of breeder male feeds didn't really change or follow a consistent trend. Even though we did get a significant value for our helper number, when we conducted our post hoc test, it basically told us that there was no difference between the number of breeder male feeds in groups that had 1 helper, 2 or more helpers, or 0 helpers. So that led to my next question of, if it's not the number of helpers that affects the breeder male feeds, is it just the presence, yes or no, of helpers? And here you can see that, yes, it was significant. When you're looking at groups that did not have helpers, the average number of breeder male feeds was a lot more than groups that did have helpers present. So moving on to my next question, how do breeder male feeds change as the contribution from other group members increase? As total number of helper and female feeds increase, which is our green line, the contribution from the male dropped. But when you're looking at groups that did not have helpers, as the number of breeder male feeds, which would be the only other group member present in the groups without helpers, as her contributions increased, the number of breeder male feeds also increased. And once again, our helper presence, yes or no, was significant in this graph. So how do total feeds vary with the brood size and helper presence? Once again, our brood size was significant in this case, and our helper presence was not. So what that is showing on this graph is that as your brood size increased, the average total number of feeds increased for both groups that did not have helpers and did have helpers. But when you're looking at groups that did not have helpers, you can see that the average total number of feeds was a lot greater than in groups that did have helpers. So what does that mean in terms of per capita feeds? And per capita feeds is just the total number of feeds that were delivered divided by the number of young that you had. And here, once again, my question is, how does per capita feeds change with brood size and helper presence? Brood size in this case was significant, and once again, helper presence was not. And as your brood size increased, the per capita feeds that were delivered also decreased for both groups that did not have helpers and did have helpers. So what does this all mean? Helper effects on food provisioning are compensatory. As helpers increase their provisioning of young, breeder males reduce their own efforts. Breeder males also worked harder to feed juveniles, especially in large broods, but males with helpers did not. And this is once again pointing to a compensatory care strategy, which is benefiting the breeder males. We also saw that even with helpers, feeding rate per juveniles decreased in large broods. And this compensatory care strategy did not benefit the juveniles at all. And this is all very interesting because previous research suggests that helpers increase annual productivity. So why is it that we are seeing a contradictory result in this experiment? Another interesting to note was that as your brood size increased, the number of per capita feeds decreased. And so it would be another good thing to look at how do reduced per juvenile feeding rates influence future survival and recruitment. Okay, so for future directions, if I were to do this experiment again, I would definitely try to sample or have a larger sample size. There were a few results that were not significant, but I think were a little bit ambiguous. By having a larger sample size, I think we can get a little bit more clarity on those results. Additionally, I think it would be interesting to look at other variables that could affect provisioning rates. Like breeding age, and the reason I think breeder age would be interesting is because of the concept of residual reproductive value, which is just an organism's potential for future reproduction. And it states that as you get older, your residual reproductive value decreases and the amount of care you invest in your offspring increases. So it would be cool to see if older birds are implementing a more additive strategy and vice versa. Another cool variable would be pair duration. Is the longer a breeding pair together going to affect what care strategy they implement? Helper age would also be a good thing to look at, especially because in this experiment, we were seeing that as the brood size was increasing, the per capita feeds were decreasing, especially for groups that did have helpers. And perhaps the reason we were seeing that trend is because the helpers were not old enough yet or didn't have enough experience to effectively take care of others. And finally, habitat quality is a good thing to look at because perhaps the reason we are seeing different provisioning rates is because there is a difference in habitat quality between groups. So why does this all matter? Well, Florida scrub jays are currently listed as a threatened species, which may be heavily influenced by the restriction to oak scrub and subsequent loss of habitat due to agriculture, suburban development, and fire suppression. And this research may provide insight on how helpers impact group member survival. Okay. For my acknowledgements, I want to say thank you to Archbold Biological Station staff for making my transition to living in Florida nice and easy, and for just taking care of me throughout my time here. I want to say thank you to the Avian Ecology Lab. Thank you to Reed and Meredith for allowing me the opportunity to work here. I am so lucky to have been mentored by both of you, and I learned a lot from both of you. I also want to extend thanks to the rest of the Avian Ecology Lab. I couldn't have asked for a better team to work with for the past 6 months. So Bird Laugh Best Laugh. I also want to say thank you to fellow Archbold interns. It was great being surrounded by a huge group of people who are equally as curious as I was. I never had a boring moment here, and I picked up so many new hobbies and knowledge from you guys, so thank you for that. And finally, I want to say thank you to my friends and family. Even though we're no longer living anywhere near each other, it's nice to know that I have your support from wherever in the country you are. And I hope that everyone will continue to follow me on my journey. Questions?
Okay, Rosa, Skylar is asking, first saying, woo, nice work. Were fledglings fed equally in groups with larger brood sizes?
I don't think I didn't see that they were. It's always a little bit, there's always one that was a little bit more dominant and kind of pushing others aside or getting all up in the breeders' faces. So yeah, there was a little bit of inequality in feedings.
Okay. Dr. Aaron David asks, can you comment on the cause and effect? Do breeder males see the helpers and cut back on their care, or do helpers seek out groups with lazy males?
I think it's the breeders are the ones that are in control of what care strategy they're implementing. So here we were seeing that in groups that did have helpers, the males were cutting back what they were contributing.
Okay, we'll do another. Could you describe the focal observation process?
Yeah, so I started off by randomly selecting my groups to observe, and a lot of it changed depending on who actually made it into the fledgling stage. Once I knew what group I was observing, I would go out and conduct my focal observation follows, where I arrive, I give them 10 minutes to kind of acclimate to my presence and go back to their daily normal behavior. And from there on, I just kept watch of where every bird was and who was delivering food to the fledglings. And it's quite noticeable when they were getting fed, just because they make this really funny begging call and wing flattering motion.
Okay, great. One last question from Jack Christie. First, he says, amazing job, Rosa. Then he asks, why did you decide to focus on the male provisioning rate as opposed to the female provisioning rate?
Yeah, when I was coming up with my proposal, I read a few papers that mentioned that the number of helpers was important to like food provisioning, but it also stated that as the number of helpers increased, the breeder male changed his efforts, but the breeder female did not change its efforts at all. So that's why I chose to focus on the breeder male in this case.
All right, great job, Rosa. Back to you, Reed.
Fantastic job, Rosa, and great questions, audience. Really loved it. So at this point, we're going to thank Rosa, and we're going to move on to our next speaker, who is Helen Nesius. So Helen, if you could share your screen. Rosa, you have to stop sharing yours. There we go. And I'd like to introduce Helen. So Helen is from Purdue University, where she worked with Barney Dunning, who's a longtime colleague of mine. She got a degree in wildlife science. And she also had a lot of diverse experiences as an undergrad, working on fish, bats, owls, various passerines. But also, I think one of the most important things is that she got engaged, even as an undergraduate, in professional service, serving as treasurer for the Wildlife Society chapter. And I think that's really important as a part of professional development. Her first post-bac job was actually really closely related to what she's doing now. She got a job with the Smithsonian Institute, working out on Santa Cruz Island in the middle of the Channel Islands, working on the island scrub jay, which 20 years ago was considered the same species. Now is a separate species that's restricted to Santa Cruz Island. And there she didn't start there until October, and then she came here in February. So she was there during the winter months, didn't really get the opportunity to do nest searching, but she did a lot of other things like banding and bleeding jays. And so she became very, very familiar with many of the activities. One of her mentors out there was Mario Pessendorfer, who has also been another one of our collaborators who works on our acorn data sets. So, so even though she didn't get to do nest finding at Santa Cruz Island, she very quickly learned how to do it here, since that's the vast majority of what our interns do. And she really excelled. So Helen also contributed a lot to a great atmosphere for this year. I mean, honestly, this is my last year of having interns, and I couldn't have asked for a better group. We keep a whiteboard in the lab with sort of our daily planning rituals, and every day I would come in and there'd be hilarious, great new cartoons written on the whiteboard. And I didn't know it until recently, but that was all contributed by Helen. So Helen chose a fairly ambitious intern project, and she really worked hard to understand it, implement the methods. And I think we've seen just a tremendous amount of growth in Helen not only as a field ecologist, but just in her confidence and belief in herself as a biologist. So with that, I'm going to introduce Helen to talk a little bit about her work on foraging strategies in Florida scrub jays.
All right, thank you, Reed. So my name's Helen Nesius, and I'm an avian ecology intern. My project focused on exploring the mechanisms behind foraging strategy variation in the Florida scrub jay juveniles. So are you a producer or a scrounger? Distinct foraging strategies occur in animals that forage in groups, and these are searching for your own food or following conspecifics to a food source. And the producer-scrounger model can be used to explain these strategies. And under this model, producers are defined as individuals that search for their own food, and scroungers are individuals that join and follow the producers to food. So what determines whether an individual will be a producer or a scrounger? In some species, dominance plays a role. So in Arabian babblers, subordinate birds scrounge more than dominant birds. And in Mexican jays, the greater the dominance difference between two birds, the more likely the dominant bird would scrounge from the subordinate bird. In other species, individual behavior and searching cost influence the producer-scrounger roles. In house sparrows, individuals did not switch strategies regardless of social and environmental context. And in scaly-breasted munias, more individuals switch from producing to scrounging when the energetic cost of searching, aka producing, increased. So do Florida scrub jays exhibit these two strategies? And the answer is yes, they do. Adult Florida scrub jays do produce and scrounge, and this was found by a previous intern, Sarah Heimbach, back in 2019. But my focus was on the juvenile Florida scrub jays. Do they produce and scrounge? And Florida scrub jays are a great model to study these strategies because they live in large groups. They have family groups consisting of a monogamous breeding pair that often have helpers, and they have brood sizes, which is the number of offspring produced during one breeding attempt, ranging from 1 to 5. So they forage in big groups. They prefer open and fire-maintained habitat with less than 2-meter tall oak scrub, so they're easy to observe while they're foraging. And the family groups also have a dominance hierarchy where their breeders are more dominant over non-breeders, males are more dominant over females. And we know that dominance can influence the producer-scrounger roles in other species. And this population in particular is highly monitored with copious recorded metrics such as weight, and they're accustomed to humans, and all individuals are identifiable via color-coded leg bands. The big question I had was because Florida scrub jays are social foragers, will they exhibit the producer-scrounger roles as juveniles? And if so, what social and environmental context influence these roles? And I hypothesized that the producer-scrounger roles do exist in the juveniles, and that variation in these roles can be explained by various social and environmental contexts. So my goal was to focus on how these 6 social and environmental contexts relate to the producer-scrounger roles of juveniles. I first looked at the presence of helpers, brood size, habitat type, whether it was open, less than 2 meters tall, or overgrown, over 2 meters tall, individual juvenile weight, their dominance relative to their siblings, and individual boldness. And while the first three were really easy to record, these last 3 required me to use, I need the help of the avian ecology lab. And so the avian ecology lab trains all juveniles every year to enter potter traps, which is this little black and white picture on the right, to attach leg bands and collect metric data. And so I recorded the trap latency, which is the number of days it took each individual to fully enter the trap as a measure of individual boldness. And I ranked each individual juvenile as fast if it took just 1 day to enter, medium if it took 2 to 3 days to enter, and slow if it took more than 3 days to enter. Once these birds are actually trapped, they're weighed. And so I pulled that weight data and then for future analysis. And I also calculated using this weight data, each individual juvenile's weight relative to their siblings. And I used that as a measure of dominance. And so I ranked individuals as 0 if they had no siblings, 1 if they were the heaviest sibling, 2 the second heaviest, and so on. In order to quantify the producer-scrounger roles in this species, I used a foraging grid. And so I trained the jays to interact with small wooden boards covering peanuts. And once I got the the J's to associate these boards with food. I recorded number of flips and joins each individual did while on the foraging grid. And a flip is just when they move or dig under a board to reveal a food source. And joining was whenever they joined or followed a conspecific to a food source. Once I got these flip and join counts for each individual, I plugged it into this equation here, and which is the number of flips minus number of joins over the sum of total flips and joins of that same individual. And that gives me a producer scrounger index for the individual. And so if the value is positive, that means it's a producer. They're doing more flips and they're doing joins. And if it's a negative number, they're a scrounger. They're doing more joins than flips. And so my experimental design, I sampled 36 juveniles from 18 family groups. I visited each group 3 times and recorded a 15-minute foraging grid trial each time. For each group, I recorded the presence of helpers, brood size, and habitat type. For each individual juvenile, I recorded weight, trap latency, and their flip and join counts while on the foraging grid. And so just to give you a quick glimpse of what this looks like, here's a video of some birds on the grid. That top bird just flipped a board and a bunch of birds are coming over to join that individual. So pretty goofy and fun to watch. All right, and so to analyze my data, I kind of bend it into a family group level analysis and an individual level analysis. So for my family group, which I had 18 groups, I calculated a mean producer scrounger index for each group. And I used an ANOVA to compare this index across groups relative to helper presence, brood size, and habitat type. For the individual level analysis, which I had 36 individuals, I used a linear regression to look at weight and the producer scrounger index, and then ANOVA for individual producer scrounger index across their ranked weight categories, which was again 0, 1, 2, and 3, and 4, and then trap latency categories, which was fast, medium, and slow. So my results, these are my family group level results, and these are for the first 3 contexts I mentioned earlier, which is the presence of helpers, brood size, and habitat type. And so first question is, does the presence of helpers significantly affect group mean producer scrounger index? So on this chart here on the right, the y-axis here, this midline is 0. And so anything above is a positive value, meaning it's doing more flips than joins, so it's a producer. Anything below is a negative value, and it means they're doing more flips than they are, they're doing more joins and flips, and so they're a scrounger. And this is a mean index. So this is just for the whole group on average, what were the birds doing? And this is not significant, but we can see that there was more scrounging happening in groups that had helpers, but the averages for these 2 groups, they weren't different significantly, but a cool trend. So then did brood size affect it? No trend here. This wasn't significant. There's not really, there's no trend in this one, so I didn't go much farther with this. And then habitat type. Again, not significant, but the average producer scrounger index overall for the group was lower in groups that were in overgrown habitat. And there was a lot more scrounging happening in the overgrown habitat overall as well. Not significant, again, but a cool trend. And then my last 3 contexts, which were weight, ranked weight within family group as a measure of dominance, and trap latency as a measure of boldness, are here. And my results were not significant for weight. So did weight significantly affect the individual producer scrounger index? There was a slight positive correlation in that the heavier the bird was, the higher the producer scrounger index was for the individual. But you can see there's a lot of spread on this graph here. So there's a lot more going on than just what weight would explain. I also have a very small R² value here. So this did not thoroughly explain what's happening. So then I looked at, well, maybe it's their weight relative to their siblings, which is a measure of dominance. And so you can see here a trend where if you have no siblings, or if you're the heaviest bird, you're more likely to be a producer, while if you are the second heaviest, third heaviest, just a smaller bird relative to the rest of your group, you're more likely to be a scrounger. This wasn't significant. I had a pretty low power too, but a really, really cool trend. And then lastly, I looked at, does individual boldness affect it? So you can see here that the faster a jay entered a trap, the more bold it was, the more likely it was to be a producer. And the more time it took a jay to enter the trap, the less bold it was, the more likely it was to be a scrounger. Not significant, but again, a really, really cool trend. So what does all this mean? So number one, juvenile Florida scrub jays do exhibit the producer scrounger strategies, just like the adults. More scroungers were in groups with helpers. And this could be just because there's more birds to scrounge off of. And then more producers were in open habitat. And this could be because jays could be spending more time with their heads up looking for predators, and also following conspecifics to a food source instead of keeping their head down and producing. So I think maybe overgrown habitat makes the jays more aware of predators and therefore want to keep their head up and follow each other. And there's also the more dominant a juvenile was relative to its siblings, the more likely it was to be a producer. And this is very similar to the Arabian babblers. And we know that also dominant jays are more bold than, dominant jays are more bold. So it brings me to my last point, where bolder individuals are often producers, while less bold individuals are scroungers. And this could be because bolder individuals are more readily approach novel food sources than less bold individuals. And that could be that could explain why more of them are producers. But overall, I had a really small sample size and a low power. But given the consistent trends I had, it'd be really cool to see what a higher sample size would do if I would have any significant findings from my analysis. Future directions, again, increase my sample size. It'd also be cool to investigate additional variables, such as not only just the helper presence, but the number of helpers in a group. It'd be also interesting to look at the number of helpers or number of territories surrounding a family group and just how dense that is. Because I often would have, neighboring kid jays come into my foraging grid at another family's trial. So I think that would kind of affect the producer scrounger rules, how dense their neighborhood is. And also it'd be cool to see who is joining who, which the Mexican jay paper did. They looked at they looked at that relationship and found that more dominant individuals were doing the joining. So that'd be cool to see that in the jays. And lastly, it'd be cool to follow the same individuals around under different contexts. So give them the trial and then trap them for banding purposes and then do the trial again. It'd be cool to maybe do the trial and then flash like a cutout of a peregrine falcon in that area and then do that trial again later and see if the presence of that predator affects it. And it'd also be cool just to follow an individual out through its life. So as a juvenile, as a helper, and then as a territory holding breeding adult, would that change? Would the producer scrounger index of that individual change throughout its life? Those are my future directions. And this is all really important because Florida scrub jays are a threatened and endemic species found only in Florida. And this all helps us better understand how the species will respond to anthropogenic pressures such as urban development and climate change. And we can construct more informed management plans with information like this. And lastly, I just want to thank the Archibald staff for giving me a great home for the past 6 months. I want to thank my Archibald peers for being super cool and nerdy and interested in the same things that I am. I want to thank my family and friends, both near and far, new and old, for just being there for me and supporting me and believing in me. I want to thank the Florida scrub jays for being adorable, beautiful, elegant, quirky, and fascinating. They're a fantastic study species. And lastly, I want to thank the Avian Ecology Bird Lab for being there for me, for being my friends, for giving me this position. They're a great group to be around. So Bird Lab, Best Lab. And with that, I'll open up to questions.
Great job, Helen. Question from Emily Jones. Do you know how these feeding strategies change as the juveniles develop into an adult, if they change at all?
I don't, and the previous research that was done didn't follow them long enough, but as they get older and kind of figure out where their dominance is, it will kind of change. They'll kind of solidify into their own roles. So I think that's something that we kind of need to explore too. Like I said, in my future directions, maybe cool to follow an individual throughout its life. So I bet it would change as it got older and fit into its group a little more. As a male, male and female features get more pronounced. So that's my answer to that.
All right. question from Beth. Did you find that single offspring were always producers or were some scroungers?
A lot of them were producers, for sure, but there's definitely a few of them that just would wait for their parents to come around and helpers to feed them. But more often than not, they were producers.
Okay. A question from Kelly. Did you see any birds pilfering caches during your trials?
Yes, that happened quite a lot. And I also had a lot of them taking peanuts out of each other's mouths while they were walking away to stash that peanut. It would get ripped out of their mouth. So yeah, that happened a lot. Or they would like take it and like, bury it just like a few inches away from a board and then, walk away. Another bird would come in and just unbury that and eat it. So it happened a lot.
Okay. Question from Greg Thompson. Do we know the sexes of these juveniles yet? I'm curious if there would be a relationship between PS index and sex.
No, I was going to add that to my future direction slide, but totally forgot. We don't have the data yet back on these Jays, but that would be super, super cool to look at once we get the data back. So to be announced on that one.
All right. One last question from Aaron David. Really cool. I wonder what the within group PS distribution looks like. For example, maybe each group only needs 1 to 2 producers. You could analyze your group data with the best producer rather than the group average.
Yeah, I didn't look into too much of the variation within the group itself besides the ranked weight, but that's a good idea.
And just a few comments, just saying both great job to Rosa and Helen. So wonderful job, both of you. Reed, do you have anything else to add?
I'd just like to thank both of these two. They did a fantastic job. And I would like to thank all the audience for attending. So thanks, everybody. Damn good.
Everyone can see you still.
I know, good.